This is a plants and photosynthesis experiment. We enter the lab, and here we have our laboratory assistant and the VRLab tablet. The introductory part contains the theoretical background, followed by the experimental section. In this section, we have the procedure, which briefly explains the experiment. We also have the tables that need to be filled out during the experiment. In the introduction, we'll talk about the Eludia plants, which take the carbon dioxide they need from water. Now, we will turn off the lights and observe if there is any carbonating in the water, indicating that the plant is performing photosynthesis. This will highlight the importance of light in photosynthesis. As you can see, in the dark, there is no bubble formation within 1 minute. However, when we turn on the light, we notice a slight bubble formation. We will count these bubbles for 1 minute. If we switch to higher intensity light, we see that the number of bubbles increases, so we will count them for another minute. After 1 minute, we will record these numbers in our VR tablet. In the dark, there were 0 bubbles. In medium light, there were around 30, and in bright light, there were around 60. The correct results are now shown. Now, moving on to the experimental section. Here we have the theoretical section and the steps for the experiment. We will also work with leaves that have little to no chlorophyll or little amount of chlorophyll. This will help us understand the role of chlorophyll and carbon dioxide in photosynthesis. I start by noting the leaf colors of the plant I took and measuring its mass. I then place it in the growth medium inside a chamber without light. Next, I take another leaf, which is greener, therefore contains more chlorophyll. I record its initial mass and place it in the growth chamber. Then, I place it in the chamber with light. I create different conditions for each leaf. The variegated leaves have less chlorophyll than the green leaves, so I measure their initial mass and place them in a chamber with light. Then, I take a white leaf with no chlorophyll, measure its initial mass, and place it in the growth medium inside a chamber with light. Lastly, I take one more green leaf, measure its initial mass, and place it in the bijou bottle with the growth medium, which is placed inside a chamber with no carbon dioxide. Following the procedure, I wait for 8 hours. After 8 hours, I will remove the leaves, measure their final masses, and calculate the change in mass. I open my timer and wait for 8 hours to finish. After 8 hours, I remove the leaves and measure their mass. I calculate the change in mass for each leaf. I take the second leaf that has light in the chamber. Then, I take the third leaf, and I measure the final mass, and I calculate the change in mass. I take the fourth leaf that had light in the chamber. Finally, I measure the last leaf and calculate the change in mass. In this way, we examine photosynthesis under different conditions, and we can see the effects of chlorophyll and light on photosynthesis.